So thank you for those of you who are joining virtually online. Yay. And I do see, I recognize a couple of the people in there. Yay. So hi and hello, come on in. So, oh, that's okay. No problem. Come on in. Uh, so Terry asked me to share a little bit about our research over the years here at the Roatan Institute for Marine Sciences of what we're doing and what we're learning about the dolphins. And so my first visit was in 2002. And then I started systematically studying the dolphins here in 2003. So there's some of the brochures if you'd like, help yourself. And so if you guys have questions or you have commentary, feel free to add for what we're talking about. But let me get this going. Before I go into all of the different research that we've done, I just wanted to introduce the Dolphin Communication Project. And we are a US nonprofit with a board of directors that's basically spread around the globe. And our mission is to promote the scientific study of dolphins and inspire their conservation. And we do this through research and education. We have a variety of different field courses as well as ecotours and other programs. And this all leads to conservation. And DCP is the umbrella under which we conduct our research and education programs and promote conservation. So we do this through a variety of different research topics to better understand dolphin communication, their adaptation to aquatic life, their social lives. And this is all done through long-term observations, which are the focus of our research questions. And we use custom-built modern technology to follow time-tested patterns and to non-invasively collect data, which informs our conservation measures. And the topics all listed on this slide, from pectoral fin contact, relationship quality, pair swim positions, et cetera, all the way down through looking at association patterns and comparing study populations, are all made possible because of the long-term observations. And these are all topics that we have studied here at the Roatan Institute for Marine Sciences, or what I affectionately call RIMS, and I know that you guys affectionately call IMS. So our education programs feature internships, field courses, classes, workshops, seminars, and most of our field courses are here. And we are actually coming up on our 10th year anniversary with Colorado State University this January, which is very exciting. We've also had students complete their senior and master's theses or doctorates with us. And we've partnered since in the last 10, 12 years with about a dozen different universities to bring students down here to experience the research that we do. So through these education programs and through our research, we want to foster the next generation of scientists. And then we aim to educate the incoming generations to be responsible environmental stewards, because in order to care for our environment, we must respect it and love it. And the only way to do that is to learn about it and to know about it. So we are on an eco tour right now. And I just like this because I love the colorful rash guards that we have. And I thank Bill and Don and John for modeling them with me in front of the lovely dolphin sculpture, which is based on the Roatan dolphins here, on 3 specific dolphins, which is pretty cool. So to study communication, you need to identify the sender and the receiver and the information. And so when I was in college and my graduate studies, I wanted to study dolphin communication, but if you've ever been in the water, it's really difficult to identify where a sound is coming from. In fact, we're built to hear where sound comes from in air. So you can either make your ears 4.5 times bigger, which is not possible, or you can split them apart 4.5 times, which we can copy that idea because you can't really pull your ears that far apart. We can copy that idea by using hydrophones scaled to 4.5 times the distance between our ears, and then cabling them into the backplate of a housing and plugging them into a camera, so that when we listen to the tapes and use headphones, for about 30 to 40% of our recordings, we can tell who's making the sounds. And then we compare those sounds with behaviors. The other 2 things you need is clear water, which we usually have here, and we usually have in our other study sites. And you need habituation. You need animals that are habituated to swimmers in the water. And we had that in the Bahamas, where I started my studies of dolphins. We have that in Japan off Mikura Island. We have it here in Roatan, but not in all other places. So to be able to study these animals underwater, where you look at inter-animal exchanges, you need these 3 things: clear water, habituation, and to be able to identify who's vocalizing. So all of our data are collected using my mobile video acoustic system. We have 4 of them that we've built, and they are all bright green because, well, we built them and that's the color I wanted them to be. And this video shows you, and this is actually from 2011 in the shallows here at Roatan at Bailey's Key. So for all of the data we collect on all the different groups of animals, we have long-term studies. We want them to ignore us. They don't often ignore us. They're very curious about the cameras. The data is collected when we're swimming with the animals, but we're trying to be non-invasive. So I don't play with them, even though they do bring me seagrass. I don't play with them. I don't interact with them as much as possible. And we document as much as we can. And we do this with a focal follow data collection protocol for all of the groups that we study, whether they are in managed care or in the wild. So we're using exactly the same methods to collect the data and to analyze the data, which means we can make direct comparisons. Yes?
Question. When you add the GoPros to the array,
the early GoPros or the early arrays didn't have the GoPros because they didn't exist. And so when the GoPros came into being, they have a much wider field of view. So I believe the first time we added a GoPro was 2017. So some of these photos are much older than that. So, but that gives us, by putting the GoPro on top of the array, it gives us a wider field of view so that we can get more animals in view and see what's going on. And 2 years ago, I added the GoPro looking back because oftentimes there are animals behind me. And I know somebody's looking at me and doing something, but I know there's somebody behind me. They're not doing that to me because their eye line is not on me. So I'm starting with the research topics with our PECFIN study. And this is from 16 years of data that we've looked at, well, more than almost 2 decades, because 16 years was a few years ago. And we started looking at dolphins in the Mikura and in the Bahamas at how they shared contact with their PECFINs. And this video that's been playing shows you what we're talking about with their PECFIN. So PECFIN to body, PECFIN to PECFIN, as they share information. The citations that are listed in blue all use data from here in Roatan. The first paper, we looked at the 2 wild groups of dolphins. The second paper, we looked at the dolphins in Mikura, in the Bahamas, and Roatan to see what was going on in terms of what we're looking at. So to sort of jump back and forth, this is the data that we collected here in Roatan. And so starting in 2003, going right through to 2018, and I still have students collecting PECFIN contact from our data from 2019 to just this past year. But for these 16 years, we had 133 hours of data that we reviewed, which is about 8,000 minutes. And you can see that the effort per year, the number of animals in the group, we had a roughly 1-to-1 sex ratio that we were looking at, and the different ages of individuals, and the number of PECFIN contacts. And we had more than 4,000 PECFIN contacts that we looked at, which meant that we could subsample the data to avoid pseudo-replication, which is statistical speak that it means we had a very robust sample size. And in fact, when I presented this information in 2018 at an animal behavior meeting, the folks there who study terrestrial animals were staggered at the size of the sample that we had for the data. So it's great for dolphins, but it's also great just for regular animals, etc. So when you're looking at stuff, come on in. So jumping back in 2010, I'm jumping in on PECFIN contact. I can go into some other stuff for you after. In 2010, we published our comparison between the 3 groups of dolphins that were our focus, the dolphins here at RIMS, at Mikura Island, which is a small island south, about 200 kilometers south of Tokyo. And the dolphins are seen within about 200 meters of this coastline all around the island. It looks like a giant muffin from a distance. And then also in the Bahamas off of Bimini, and which is south, like right about here is where Bimini might be. And then the White Sand Ridge up in here where we see Atlantic spotted dolphins. What we found is that this behavior, PECFIN contact, is very conserved in that all of the dolphins that have been studied use it exactly the same way. And this is confirmed with the literature. So the dolphins in Australia that have been studied at Monkey Maya use it the same way. So we found that contacts and rubs and touches were used the same at all 3 of our sites, with one exception. At Mikura Island, we saw no juvenile groups, So we saw no contact just between juveniles. And we also saw no adult females with juveniles by themselves. So no single adult female juvenile groups. But probably because infanticide was practiced and probably still is practiced at Mikura Island, where adult males will single out a female, not her first calf, because she's usually with an experienced mom and her calf, but a female she's on her second or her third calf, and the males will single her out and try to get the calf and kill it and probably bring her into estrus sooner. And so I actually observed some of that. It was documented. Calf carcasses were found. And so this is something different. This is observed there at Mikura, but not at these other two sites here and in the Bahamas where we've observed this. And so the maternal strategies for calf rearing seem different between Mikura Island and the others. And so that's why we see a little bit of difference in the pec fin contact probably. So one of our main hypotheses was looking at, is pectoral fin contact used for social bonding? You know, we use hugs and handshakes, we pat people on the backs. And so we found no difference really in how pec fin contact was used between the groups. But then look at the difference in the amount of data. When we're looking at pec fin contact, this was for just 10 years, we had 3,000 pec fin contacts from 108 hours. And then of course we added another 1,000 pec fin contacts in another 33 hours of data. But compare that to Japan and to Mikura, or to Mikura Island in the Bahamas, where we only had about 450 pec contacts over 7 years and 139 pec fin contacts over 4 years. And that's because the return on effort from those two study sites is so much greater. I can tell you there are numbers that I will never forget. For the 4 years it took me to do my PhD work in the Bahamas, I spent 1,980 hours looking for dolphins. And from that, I had 40 hours in the water. And from that, I had 20 hours of video. So it's a 1% return on effort. At Mikura and in Bimini, we have about a 15%, 10 to 15% return on effort in terms of looking for animals and having the video. So we have about an 80% return on effort here. And since we found no differences, for the rest of our hypotheses, we focused here on RIMS, on the dolphins here, to look at what was going on in terms of how dolphins use these behaviors. So with support also from the literature, we confirmed that pec fin contacts are used for social bonding. And this is especially true among young animals, especially true among young males. And this was seen in a couple of other locations in Australia, also off Florida for what they can see for the pec fin contact. You have other contacts going on between Tank and Stan, two young males. This is about 5 years ago between these two. They like to mouth each other, not just, others in the water. And what we found is that pec fin contact wasn't really used for tension reduction as much as social bonding, and it wasn't used for social status between individuals. But it is used, they will interact with each other and exchange pec fin contacts, especially with individuals that they're not kin with. So they'll share contact, interact with one another, and then do that over the years. Yes.
So don't you think it's really so important that the relationship, all these animals, who's kin, who's not,
yes,
how old they are, what sex they are, if they're, like I said, kin
yep,
or not kin, and also the fact that the animals are all kept together like a natural pod, so
yes,
you see the natural behaviors occur in here, just like you would see in the wild.
Yes,
have so much more background information.
Yes. The fact that I can sit down with you guys, with any of the trainers or you guys, and know history, know who they're related to, but also medical history. If somebody's having an off day, they may not want to interact, and that feeds into it. Whereas if you don't have that information, you don't know what might be factoring into the behavior of these animals.
So in the Bahamas and Mikura Island, you don't know.
We don't. Nope. And actually, as I go through, you'll see more of that. So here we know kin. So in this video, you see Bailey with Tank when he's 3.5 months old, and Callie, her younger sister, is with them. So what we've seen from the data is that when a female has her calf, within that first 1 to 3 months, a relative can hang out with her, or maybe a very good close friend, but mostly we've seen relatives. But then when the calf gets older, when Tank is almost 1.5 years old, Tilly's hanging out. And so this factors into what I'll chat in a bit about alloparenting as well, and practice for the young females with other calves, but also in seeing how each female handles their calves. So we've even been able to look at that with pec fin contact. But the thing that you see with this video is there's very little pec fin contact. So from all of that data, from that 16 years, we had the 4,300 pec contacts, 3,500, almost 3,600 were between non-kin. And then we had, a little over 765, 17% of those contacts were between kin, half of those between mom and calf, and half between siblings or aunts, because we have siblings and aunts here, and we know that. You'd need DNA, actual DNA sample from the animals in the wild, which is not easy to get to. And so I'll show you that in a sec as well.
No, but I mean, if you see this behavior, you know who is who and who's related
Yep.
to who. And then you see this in the Bahamas, you see it.
Yes.
You can sort of kind of.
Oh, yeah.
That they're.
Most definitely. Yep. That's the neat thing about what we've done. I still, when we submit papers for publication, that we've published on this in the early 2000s. To me, there's no difference between managed care and the wild, as long as you have a good facility with choice and control and good welfare of the animals. So that the data that we have from here, there's no difference when I'm looking at it in comparison. And I have reviewers who are like, no, you have to prove that. I was like, here, take these papers. We've already done that.
Yeah.
And this is where the stuff that we learn on animal, technical term, the stuff we learn on animals in managed care helps with the wild and vice versa. So they need to work hand in hand. So the other thing, some of the research and there are some research topics that can only be done with managed care and some that can be done with both areas. And this video was supposed to play, but this is Ellie and Gizmo. And this, you'll see coming into the side is Alita, another adult female. So you have a couple of pec fin contacts. Of course, we thought that there was not much pec fin contact at all. And then we had several other young animals and we see pec fin contact being used in a variety of different ways, which I love watching this video of Gizmo because I think she's just a hoot. She does. Yeah. But watch. Yeah. Alita comes in and Alita and Ellie start exchanging pec fin contact and Giz comes in a little bit. Well, over the years, when we've been able to look in more detail at the pec fin contacts, not only do we see that young males start using pec fin contact when they're juveniles or even as calves, and then they continue as they get older and they use that as adults. We saw a difference. Young females with other young females don't use the pec fin contact as much, but then as the females age, and they're adults, they use pec fin contact a lot. And one of the things that we had been coordinating with Terry about is the use of their pec fin contact seems to correlate with reproductive condition or reproductive status. So females that have CAS and are nursing share more pec fin contacts. Females that are pregnant share more pec fin contact. And then females that don't have calves, young calves, or aren't pregnant or et cetera, their status, they will share more contact, at least what we've seen in some of this stuff here with the number of females that we've seen with calves, which is actually pretty exciting. And what this says to us is that, one, other research has shown into signature whistle use and long-term social memory that dolphins can recognize their kin. So they probably don't need to use pec fin contact when establishing or maintaining those bonds, but they need to use pec fin contact with individuals that they're not related to, because that gives them a way to make sure that those relationships are strong and tight between individuals. So we could take our study of pec fin contacts a little bit further, and we could look at maternal style, and then we could also look at calf-initiated pec fin contacts. And the results are two papers. If anyone's interested in them, I can share them with you. They're on the DCP website, but we focused in on, for these papers, on 11 females. We now have a couple more that we can add in, but 3 of those females were second generation moms. So it's not just looking at the individual behaviors and the exchanges, but we can now look at generations of individuals and follow individuals over years developmentally because we have that data. We know their birth dates. We know when they're born. So we know how old they are. We don't need DNA data to confirm who's related to whom. In the wild, you do, because adoption exists. There's a couple of instances that have been confirmed that other dolphins may adopt. So you either need to collect skin or feces samples to confirm DNA. And there's very few locations where you have dolphins that are habituated to human swimmers, where you can get close enough to observe what's going on. So this is why this group is so excited, because exciting to me is not just what we've been observing, but we can look at the detail and we can coordinate with the training team to get more information about the individuals that we might not know and we wouldn't know from the wild. So from our review, and for this, we did look at pec fin contact, even the limited number of pec fin contacts exchanged. We confirmed that each mom has a different style, but each mom is consistent in how they treat or behave with their male and female calves. They don't treat them differently. They might keep the male calf longer than the female calf, but they don't use behaviors differently with them. And when we looked at calves and how they initiated the pec fin contact behavior, by and large, they shared more with their mom than they did with anybody else, but then with kin and non-kin equally. And they initiated more pec fin contact with others as they got older. So we looked at each calf as a 1-year-old, a 2-year-old, and a 3-year-old to see how they were exchanging contact with age and if that would change. So the younger calves definitely did more with mom, but then as they got older, they branched out and shared more contact with other individuals as well. Because this is just pec fin contact, we're seeing there's a lot of other like body-to-body contact and other exchanges. And one of my students who looked at the calf-initiated pec fin contact with a few individuals for her undergraduate thesis, she's now looking at all contact. This is Savannah Terry. She's looking at all contact behavior with calves initiated, and she's looking at the last 7 calves and over like 6 years of data to look at how it changes with their age, and it's all different contact behavior, as well as the pair swims they use with mom, which is pretty neat. So she's working on her stats right now. Yeah. So for our studies of maternal style and calf-initiated behavior, we don't actually have enough data to do a comparison with the animals in the wild. One, because when we sight the animals in the wild, there are sometimes days, if not a month, between sightings. So you can even miss when a female actually has a calf. And if the calf dies, you won't even know that she had a calf. But also, we don't have DNA from these animals that we study in the wild because they're part of swim programs. And a lot of the boat captains don't want any invasive methods to collect data on them, which I agree with, because you have this trust that's been built up. So there's other non-invasive ways to collect some data, but we don't yet have enough data to do that comparison with any of our groups in the wild. So I decided for this one, I was just going to show the video that we have. And this is a really exciting case that we had published. The video is available on the Aquatic Mammals Journal website. And it talks about alloparenting. Yep, Gracie. And we do label them because we share this on the website. But Polly, she had her first calf. She was 12, right? 11 or 12?
Yeah, she was still.
Yeah. But she practiced a lot with almost every other young calf. She would alloparent what we saw with Tank and Sandy and Woody and Trixie and Gizmo. And in this particular situation, she had Trixie. And I got to watch a couple of times where Gracie watched, but let Polly take Trixie. And Trixie, even you'll see a couple times, Trixie tries to nurse from Polly. So Polly is getting used to what it might feel like to have a calf nursing. There was one instance where Polly had Trixie and Gracie was watching, and Polly got distracted by me and turned away and didn't pay attention to Trixie, and Gracie swooped in and took Trixie away. Polly got a little irritated, and I told her it wasn't my fault. She was the one that lost attention. But there she is. Gracie is nursing, trying to nurse from Polly, where Polly had no milk. So, but it's really neat to watch the more experienced moms like Gracie watch when you have somebody else alloparenting with their calf. And we have video of Polly doing this with Sandy, with Tilly off to the side. So this is practice. And we got to see it firsthand here because the younger females get a chance to practice with a calf in a safe environment with mom watching. And so they're learning and developing the skills that they need to be a good mom. And so we were excited we were able to put this into a special issue of Aquatic Mammals. So if anyone's interested in this, happy to share it. But this gives insight into what we see in the wild for females and how they might need to develop some of their skills to be good moms and to have those calves out there in the wild in different spots. So the most recent study that is in review right now, because I got a little irritated with there's several facilities in different countries that they ban breeding and they don't allow breeding, which to me is not, I don't agree with that. Breeding is natural, normal behavior. And then having an offspring is natural, normal behavior, as we've seen numerous times here. So I, with my colleagues, figured out how can we assess this? How can we say that offspring are important? How can we show with the data that offspring are important to a group, to a social group? And so what we decided to do was to look at the number of calves we had present and the group size, the activity level of the group, the spreading of the group, and the various other aspects of these groups that we saw here. And so we used data from 2021, 2022, and 2023 because we had a good representation of calves of 1-year-olds, 2-year-olds, and 3-year-olds. And so the C1, C2, C3 are the year old that the calves are. And on the right, you'll see the calves of the same ages, or same calves at different ages. And of course, pink means they're female, and the green, because I didn't want to do blue, are the male calves there. And you can see we have them at different ages, so we can do comparison and look at how the social group might change when you had different aged calves. And so we had about 10 hours of data from each session, or for total for when we were looking at the data. And we had equal amounts of video from each of these trips for the data to have for comparison. And we selected these sessions to maximize the amount of groups that we had and the number of calves. And so in total, we had 21 bottlenose dolphins across these, 12 females, 9 males, with 7 calves in total. And we found that, yes, definitely, everything correlated with the number of calves and the group size. So we had larger groups, we had more interaction, more activity, tighter spreading with the groups, and more overall activity with the groups. So we were able to show that, yes, offspring are important to social groups. And so these are the mom-calf pairs that we used. We have Gracie and Trixie in 2021, Tilly and Sandy, and the dates are the years that the calves were born for comparison. So we used Trixie, Gizmo, Woody, and Cammy as 1-year-olds, 2-year-olds, Buzz and Rocky, and Sandy as our 3-year-old, and then overlap in a couple of those individuals for 2-year-olds and 1-year-olds. But we have as moms, Gracie, Tilly, Callie, Polly, Ellie, Alita, and Maury, and Trixie, Sandy, Cammy, Buzz, Rocky, Gizmo, and Woody as our calves. So I was able to get them all on there, although I had to move some of the pictures around for some of that. And because this is a little bit about the science we did, I did have to show, one graph. I know, John, you would prefer me to have all videos, but we have some of this. And groups with a calf, we had almost 70% of the groups with a calf were much larger groups. And we did have groups with no calf, but those were usually one individual, occasionally with 2 individuals, and rarely with 3 individuals that were not calves, juvenile groups usually that were with one another. And you can see the examples of the different group sizes when we'd see the calves in the groups. I could spend more time on this because I find this a fascinating topic, but I'd be happy to share the paper that's in review right now, and hopefully it will go out sooner. And we have been sharing this far and wide with a couple of different organizations where they're dealing with politics right now, where governments are trying to put laws into place about breeding. So we're trying to counter that with some of these data. So jumping to a different topic, stepping a bit away from pec-fin contact as well, is association patterns over the years, whether you're looking at terrestrial animals, avian animals, or aquatic animals, has always looked at the time spent together. And this is done either with surface observations or underwater observations. But looking at time together doesn't necessarily tell you about the quality of the relationship that 2 animals might have. And so Mano Temelin, who usually comes down here and helps me out with data collection on our field courses, for her master's degree, wanted to look at relationship quality. This was a study that was originally done with chimps and ravens. And so she took the method that was used for a social terrestrial animal and an avian social animal and applied it to an aquatic social animal to look at the quality of the relationship between individuals. And that is based on looking at 3 components, which are the value, the security, and the compatibility of each pair of individuals in a group. So the value is what individuals gain, what's beneficial to them from that interaction. The consistency in the partner's responses over time is the security of that relationship. And then you can look at the personalities and the social exchanges between individuals to give you an idea of their compatibility over time. And looking at how individuals interact with one another allows you to assess the quality of their relationship. And we do that by looking at 8 behavioral variables. And this is a comparison of those behavioral variables between chimpanzees and ravens and dolphins. And so those that are in red are value behaviors, blue are security behaviors, and green are compatibility behaviors. And so there's overlap between these different behaviors, which says to me there's a lot of consistency and conservation in the behaviors that are used for these different functions. So grooming in chimpanzees is similar to preening in ravens, which is similar to rubbing in dolphins. But it may value so the preening in ravens is more valuable, whereas it's compatibility in chimpanzees. And the rubbing is also compatibility for the dolphins. The one behavior that we weren't sure of because it seemed to fall, and I should jump back. For the dolphins, Minot looked at the dolphins here at RIMS and the spotted dolphins in the Bahamas. And the one behavior that fit one place in RIMS and a different place in Bahamas was pair swim position. So it was in value here at RIMS, and it was security in the Bahamas. And that's looking at infant and echelon position, and then 5 other pair swim positions that Minot had identified. Now, pair swim positions, looking at infant and echelon. So echelon is where you have the calf up on the side of the mom, and then this is infant position where you have an individual this is called the genital area right here. It's halfway between the dorsal fin and the flukes. And those are both energy saving, hydrodynamic benefits for the calves. They're also camouflage for protection. And it gives the calves, they've been confirmed with colleagues who studied physiology that these are energy saving behaviors. So the calf doesn't have to expend as much energy when they're in echelon or in infant. And echelon is usually when the calves are younger, and infant as the calves get older, they can be in that position because they're close also to the mammary glands. So what we started seeing is these positions, we see them in juveniles, in subadults or teenagers, and adults. And we see them of both sexes using these behaviors. But then also, Minot confirmed these other pair swim positions. And excitedly, the paper was just accepted on Monday for publication. Yay. But we are looking at infant position and echelon, infant in the upper left, echelon in the lower right, but also parallel, staggered parallel, or stacked, the various different positions. And there's a poster up in the back over there if you want some more as the cliff notes, and then the paper, happy to share. What we showed is these behaviors, these pair swim positions are important socially. We haven't yet figured out, we're still talking to Shane about this for exercise physiology, about how to look at this energetically. We haven't yet figured out how to do that for the different positions, but these positions and where animals swim seem important socially to them. Because we'll see, I remember watching Maury in infant position to Paya and also to Hector and swimming that way. We also see sometimes with moms and calves, Polly likes to be in infant position to Cammy, which they switch around when she nurses, but you have that different positioning. So these are the different types of positions. And this first paper really sets the stage and gives the definition of them. But now we're looking at these positions with contact, with object play, with other behaviors that we're also documenting. So jumping into another topic, 2 topics, cognitive research. We've done creativity studies here, and we're also working on a reading study. And for some research, it's just not possible to do this with animals in the wild because you need to coordinate and ask them questions. You want to ask the animals to participate, not just be observationally included, where all the previous research I was chatting about, we actually collect the data and try to be non-invasive and just observe. With these studies, we're actually asking the animals to participate. So you have the creativity, which is it's innovate or create, Terry, with the Okay. And so as you can see in the upper photograph, Terry asks, I think that's Lenka in that photo. They give the cue and ask him to do something new and something that's different from what he previously did. And this allows us to use a Torrance test to look at originality, fluency, flexibility, and elaboration in the behaviors that they provide. There is, I'll show you in a second, we do have a nice 12-minute video about the creativity study on the DCP website, but I also wanted to keep it brief tonight and not share the video since you can see it there. And this short video down here, that is Bailey. And we are working on how they might process 2D information. So with this short video in the reading study, you present the behaviors, certain behaviors are matched to a two-dimensional picture, black and white picture, and the dolphin learns to do those behaviors. And once they have all those behaviors and they're doing them consistently, then we're going to present two symbols at the same time and see how they process. Do they read left to right, right to left, top to bottom, bottom to top, or do they do them at the same time, which helps us identify how they're processing information. So that's to come. That's something that's still ongoing. These studies are also enriching. I firmly believe these studies are very enriching to the animals because they choose to participate or not. This is the creativity video on our website. You can see also was a dolphin webinar that we did. We have all of our webinars on the website, a couple of papers that are there, a few more that are in press or in review. And we're taking the creativity study one step further. And I have a student who's going to be looking at she is looking at object play. She's at Nova Southeastern. And she is looking at episodes of object play among the dolphins here at Roatan and the Bimini dolphins, looking at both to compare them, to see how they play. What do they do? What are the sequences that they do with these items? And then who do they do it with? So this is a video that I love. It's from a while ago. And it's Tank and his palm frond. At one point, it looks like an air guitar for him. I just think it's pretty cool. And others, Buzz and Rocky come in, and Sandy comes in, and they all try to get it, but he keeps it with himself. And so using object play and using this natural behavior, we can look at what do they do with these objects? Do they grasp them? Do they share them? Do they steal them? Do they pass them around? We've seen them play with these items, seagrass, palm fronds, occasionally, pufferfish and other animals. They'll have them on their rostrum, on their dorsal fin, on their fluke, pass them back and forth. That's a various amount of elaboration. And so that can give us insight into how they are naturally or spontaneously creative with some of these actions and some of these behaviors and these items. And then we can compare that to when we work with the animals and ask them to be creative. And I think they have a lot of natural items here, and so they just are creative, in my view. Although I'm not really a fan of when I'm the object that you're playing with. So throughout the research that I've done, I also use comparative research. So some of the stuff that Minot did with relationship quality, she was looking at behaviors across taxa. So looking at birds and chimps and dolphins and seeing that there's a lot of comparison there. 3 years ago, almost 4 years ago, we started studying killer whales at SeaWorld and just recently added Loro Parque to that topic, using exactly the same protocol and methods that we're using to study the dolphins here. Although I promised I would not get in the water with killer whales. So we're just using the video of killer whales, the underwater and surface video. And we've recently, this summer, gotten video from 3 different groups in the wild. And so what we're seeing is that large and small dolphins use their pec fin contacts the same way. And when I say large and small, I mean bottlenose and spotted dolphins for small dolphins and killer whales as the largest dolphin for the large dolphins. The male killer whale pec fin is giant, so it looks a little different to the bottlenose dolphin pec fin, but they use it the same way. We also see them using pair swim positions the same way. We see them socializing and using object play. We have this beak to genital behavior that they're using the same way. All very interesting behaviors, and it's just very consistent. And it doesn't matter whether they're in the 2 facilities or they're in the wild, they're using these behaviors the same way. So I could, as you guys, most of you know, I could stay here and talk for hours and hours, but you can gain more information from the DCP website, from our dolphin pods, our podcast, from our deep dives and our webinars and our dolphin lessons. We're recording this presentation today, so happy to share it and put it out and make it available to individuals who might not have been able to be here. And of course, we do programs. So we have openings in field course and our eco tours and field courses. And that's what we're here right now on. And then we can be reached by social media. Thankfully, Kel oversees all of our social media, because if she didn't, we wouldn't have it. And we have our website with lots and lots of information, and we do a blog that we're posting on regularly as well. So I don't know, does anybody have any questions? Okay. Yep, no problem. Thank you. Cal, were there any questions from anyone?
Hello, we didn't have any questions. There was a comment about when you were talking about calf adoption in the wild and if that has occurred at RIMS.
I'm going to stop the share so we can actually go into this. And let me just go out of this. Yay! Quit the PowerPoint. There we go. You guys can see? Yeah. Okay. Hi, everybody. That's not a question I can answer. Has there been, I think one case was it? Somebody asked about has calf adoption happened here at RIMS? And I remember one case at least, and that was Mrs. Beasley and Champ, right? Yep. So to whomever asked the question, Maury did not like Champ, didn't want Champ, didn't. Yeah, I think he was the first one, and she was like, oh, yeah.
Had the baby, and Casey was Maury's primary trainer. I was Gracie's, and Gracie had Tilly. Tilly was a year old, was already eating fish, and Champ was born at 4 o'clock in the afternoon. By the time it was dark, Maury was nowhere around. We thought she had him.
Okay.
And in fact, it was Gracie that had him. They looked so much alike. When it was getting dark, we mistook Maury
for
for Gracie.
Okay.
But then it got dark and we couldn't see any because we didn't have the lights on the facility at the time. And in the morning, Mrs. Beasley, who also had Vin, had Champ and never let Champ go.
Okay.
Kicked Vin off.
Yeah.
And took on Champ.
So yes, it's happened. And I think it probably happens more in the wild than we realize.
Yeah.
And I learned from you guys, when you have a first-time mom, she doesn't really show until near the end. But in the wild, we don't see, and you can confirm this, Cal, we didn't see moms all the time, especially first-time moms. And if you saw her maybe every 4 or 5 weeks in the wild, if she had her calf but lost it, you wouldn't think that she had a calf, and so we know here when they have their calves because you can see them. And I believe when I was in graduate school, there was a study, Jay Davis was looking at toxicology of the stranded animals. And he had the idea that most females lose their first calf because they, one, are inexperienced, and two, can shunt all the toxins to the calf. So it's got 2 strikes right off the bat when it's born and doesn't quite make it. I think that, to add to that, I think that may also be why I think females live longer than males, because they can get rid of some of those toxins and it doesn't, and I was asking Shane about that, and he's sent me a couple other citations because I wanted to look some more of that up to get that. But that seems to be the case where I think we've underestimated or overestimated the age at first reproduction for a female because we didn't see it in the wild. So.
Oh, that's the big thing. You can't see them in the wild like you can see them here.
Exactly.
And that's why I think this work is so important.
Yep. 100% agree.
We have another question, Kathleen, and then if you wouldn't mind, for the sake of the recording, we can just barely make out what other people in the room are saying. So if you want to summarize any of the key points that are added, the recording would appreciate it.
Okay.
The next question is, do dolphins ever deceive each other to try and get a choice object from another dolphin?
Do dolphins ever deceive each other? I think you guys have more experience with deception, maybe, with dolphins deceiving one another. I haven't, usually when I'm working with them, I'm just watching how they interact with one another. And I actually have, yesterday afternoon, I watched Trixie tease Buzz with a piece of seagrass. She held it and dangled it in front of him. So do I think there's teasing going on? Yeah. Do I see it a lot? No. Just because I don't know if I know what I'm looking for. I mean, we're now able to see the subtle intricacies where we can see the subtleties of how they interact with one another and know, a subtle shift and a look, it means something that we're not used to the subtlety of that. I mean, I watch all of the trainers with the animals and and both eyes can see something different. So you can have one eye on you and one eye doing something over here. And it's like, nope, nope, nope. You need to focus on this. And I can see that when I'm observing them. It was a little freaky the first time I saw Stan like pooch out his eye and stare at Rocky with one eye and then look over here at something else. It was like, yeah, I was like, it's sometimes the eyes are a little freaky. So, but would you guys know of any deception, if it as such as it were, or any like interaction with animals?
You have anything to say?
Yeah, it's not fun.
You mean like when they beat each other up to steal others?
I agree,
You could go maybe talk into the.
No, that's okay. I can repeat it. Yeah,
Okay.
thanks.
Rita was in the water columns, kind of cruising along, and Coach Esteban come up behind her. As soon as he got behind her, she hit him in the throat so hard I thought that she killed him.
Yeah.
So
They both took off for a while, but then they both showed back up, so he didn't die.
that's good.
But
And
that's.
she's kind of faked him out, like, come on, ready for you. And she got close enough, she really laid into it.
Yeah, so I think Christine heard that because I see the shocked emoji there. But and I've seen that. I saw we have a footage of two juveniles of this one in Japan where one is kicking the other one and it comes out of nowhere and it's the up kick and you think, oh crap. And it looks like it hurts. I mean, I don't know what they did.
Though. I mean, it would be like me going like this, you stop it, you know what I mean?
Yeah.
They're so tough, but when it comes to objects, if Buzz really wanted that from.
He would grab it.
Gracie, he would have just taken it.
Yes.
He wouldn't have let her deceive him.
Yeah.
It doesn't go like that.
No. But I mean, that was, I was sitting there watching it going, oh, she's, he's
Yeah.
like trying to grab. I was like, okay, well,
But it was just tolerated.
yeah. I've seen them try to take the little puffer fish from each other, which I feel bad for the puffer fish because it's like this big and they never survive.
Yeah.
The two Gizmo yesterday morning, Gizmo swam up to me and was like, she had a puffer on her tongue. She just
I love that.
I was like, oh yeah. Puffer wasn't moving though. Yeah. So.
Yeah. And that's I'm picturing even human toddlers who well, my children are not toddlers anymore and they still do it. Like one is playing with something and then the other one wants it. Like you didn't want that before the other one was playing with it. I see that in friends' dogs. Like you had no interest in that toy until
Yep.
the other dog went for it. And whether or
We
not
have
there's
people
any
that does that.
of that goes on with their object play.
Yeah. Yep. Most definitely. Cool. Any other questions from you, Cal? Or oh yeah. Beagles do that all the time. Yes. We have Hazel has her toy that used to be a pheasant. And now it's just a brown ball that our neighbor called a potato. And we're like, no, that's pleasant pheasant. It has no wings, no head, no tail. And if Baloo looks at it from across the room, Hazel grabs it and it has to be hers. Yeah. So yeah. Cool. Well, any Any other stuff?
No, that was great. Thank you
Awesome.
so
Thank
much.
you.
Thanks for everybody joining.
Yes,
I just
thank you.
think this is just so important that we talk about this every chance we get,
Yes.
because, you know, our industry, our science, our research is being attacked.
Mhm.
And we're so busy doing the work, we don't take the time to talk about it enough.
Yep.
And we need to do this more often.
I agree. I agree. Well, this, I'll make sure you know, we'll coordinate, we'll have this available so that people can listen to it more and go from there. So thank you.
Thank you guys for coming.
Yeah,
It was interesting.
I'm going to sign off, Cal.
Yeah.
Thanks. Good to see everybody.